The human body is made up of trillions of cells. Every day, millions of them divide to help us grow, repair damaged tissues, and replace old cells. This is the cell cycle. The cell cycle consists of two main stages, interphase and the M phase. Interphase consists of G1, S, and G2, where the cell grows, replicates its DNA, and prepares for division. The M phase includes mitosis and cytokinesis, where the cell divides into two genetically identical daughter cells. During G1, the cell begins to grow in size. At this stage, the cell contains 6 chromosomes. Each chromosome consists of a single chromatid because DNA has not yet been replicated. The nuclear membrane protects the DNA inside the nucleus. The cell also produces proteins and organelles. The centrosome begins duplicating, and each centrosome contains a pair of centrioles. By the end of G1, the cell has grown and is ready to enter the S phase. Next, the cell enters the S phase, where S stands for synthesis. During this stage, the cell replicates its DNA. Each of the 6 chromosomes is copied. Even after DNA replication, the cell still has a diploid number of 2n equals 6, meaning it still contains 6 chromosomes. The difference is that each chromosome now consists of 2 sister chromatids, which are joined together by a centromere. Next, the cell enters the final stage of interphase, which is G2 phase. During G2, the cell continues to grow and produces additional proteins and organelles needed for cell division. By the end of G2, the centrosomes have finished duplicating, and each centrosome contains a pair of centrioles that will help form the spinal fibers during mitosis. Once these final preparations are complete, the cell is ready to enter prophase. Next, the cell enters prophase, which is the first stage of mitosis. During this phase, the chromosomes become shorter so they can move easier. Each chromosome has 2 sister chromatids joined at the centromere. The nuclear membrane begins to break down, which allows the chromosomes to move freely within the cell. The 2 centrosomes move to the opposite sides of the cell, which begin to form and prepare to attach to the chromosomes. The cell is now ready to enter metaphase. Next, the cell enters metaphase, the second stage of mitosis. During this phase, the 6 duplicated chromosomes are lined across the center of the cell. As the chromosomes line up, spinal fibers attach to each chromosome at the centromere. Each chromosome still consists of 2 identical sister chromatids joined together at the centromere. The cell is now ready to enter anaphase, the third stage of mitosis. During this phase, the centromere is split, allowing the sister chromatids to separate. The spinal fibers shorten, pulling one chromatid from each chromosome toward opposite sides of the cell. Once separated, each sister chromatid is now considered an individual chromosome. The cell is now ready to enter telophase. During this phase, the chromosomes reach opposite sides of the cell. New nuclear membranes form around each set of chromosomes, creating 2 identical nuclei. Since this is an animal cell, a cleavage furrow begins to form as the cell membrane starts to fold inward. Finally, the cell enters cytokinesis, the last step of cell division. During this stage, the cleavage furrow closes in until the cytoplasm is completely divided. This separates the parent cell into 2 genetically identical daughter cells. Some cells enter the G0 phase instead of continuing through the cell cycle. In G0, the cell leaves the cell cycle and stops preparing to divide. Instead, it carries out its specialized function.